Antigen receptor signaling is enhanced by co-receptors that bind the same ligand. Optimal signaling through the T-cell receptor complex occurs only when it clusters with the co-receptors CD4 or CD8. This is why most T-cells with MHC class II restricted T-cell receptors express CD4, which binds to the membrane proximal domain of MHC class II molecules, whereas most T-cells with MHC class I restricted T-cell receptors express CD8, which binds MHC class I molecules. Expression of the appropriate co-receptor means that the peptide MHC complex bound by the antigen receptor can be simultaneously bound by a co-receptor molecule. About 100 identical specific peptide MHC complexes are required on a target cell to trigger a T-cell expressing the appropriate co-receptor. In the absence of the co-receptor, 10,000 identical complexes, about 10% of all the MHC molecules on a cell, are required for optimal T-cell activation. This density is rarely, if ever, achieved in vivo. Aggregation of the T-cell receptor with the appropriate co-receptor helps to activate the T-cell by bringing the LCK tyrosine kinase associated with the cytoplasmic domain of the co-receptor together with the ITAMs and other targets associated with the cytoplasmic domains of the T-cell receptor complex. One of the principal targets of LCK in T cells is another kinase, the zeta chain associated protein, or ZAP70, which is important in propagating the signal onward. B-cell receptor signaling is also enhanced by aggregation with a co-receptor. The B-cell co-receptor is expressed on mature B cells as a complex of the cell surface molecules CD19, CD21, and CD81. One way in which this complex can be co-ligated with the B-cell receptor is through the recognition of an antigen that has activated complement. The CD21 molecule, also known as complement receptor 2, CR2, is a receptor for the C3d fragment of complement. So antigens, such as pathogens, that have activated complement directly or through the activation of antibody can cross-link the B-cell receptor with CD21 and its associated proteins. This induces phosphorylation of the cytoplasmic tail of CD19 by B-cell receptor-associated tyrosine kinases, which in turn leads to the binding of SRC family kinases and the recruitment of a lipid kinase called phosphatidyl inositol 3 hydroxide kinase, PI3 kinase. Co-ligation of the B-cell receptor with its CD19, CD21, CD81 co-receptor increases signaling 1,000 to 10,000 fold. The role of the third component of the B-cell receptor complex, CD81, is as yet unknown. And that's antigen receptor signaling is enhanced by co-receptors that bind the same ligand. Have a great day.